Hello, let me ask you something. Have you ever thought about how your body grows, or how a cut heals on your skin, or even how some diseases like cancer start? The answer to all of those questions begin with cells and how they divide. Every second, millions of cells in your body are dividing, making sure your body grows, heals, and functions properly. And they don't just split randomly. They follow a specific process called the cell cycle. Today, we're diving into the life story of a cell, learning how it prepares to divide, when it divides, and how your body keeps this process under control to keep you healthy. Now, what is the cell cycle? The cell cycle is the series of events that a cell goes through as it grows, prepares to divide, and then divides into two new cells. It's basically the cell's life plan, helping it do important jobs like repairing tissues, helping you grow, or replacing old cells. It ensures that each new cell has exactly the same genetic material or the DNA as the original one, so your body stays healthy and balanced. The cell cycle has two major phases. The first is the interphase or the preparation phase. This is the longest part of the cell cycle, about 90% of the time. The cell is not dividing yet, but it's very active, growing, copying DNA, and preparing to divide. Think of it like studying and packing before a big move. The second phase is the mitotic phase or the M phase. This is the division phase. This is when the cell actually divides. Includes mitosis, division of the nucleus, and cytokinesis, which is the division of the cytoplasm. The result? Two identical daughter cells. The cell cycle has four stages. The first one is G1 phase or GAP1. The cell grows larger and makes new organelles and proteins. It performs its natural functions like a skin cell doing its job. The cell decides whether to divide or to stay the same. And there's also a checkpoint that checks for size, nutrients, and DNA damage. The second phase is the S phase, or the synthesis. The cell copies its DNA so that both new cells will have a full set of instructions. This is critical. Without accurate DNA copying, the new cells won't function properly. Now, the third phase is G2 phase or GAP2. The cell continues to grow and makes final preparations for division. It checks the DNA for any errors and begins forming structures needed for mitosis, like spindle fibers. Another checkpoint makes sure everything is ready for mitosis. And the last phase is the M phase, which has cytokinesis as well as mitosis. The cell divides its nucleus, which is the mitosis, and then its cytoplasm, which is under cytokinesis. Mitosis has four phases: prophase, metaphase, anaphase, telophase, which will be discussed later on. After division, two identical daughter cells are formed, each with the same DNA as the parent. So, simple analogy, think of the cell cycle like baking two identical cakes. In G1, you gather and prepare ingredients. For the synthesis phase or the S phase, you copy the recipe. With G2 phase, you are double checking everything. And with the M phase, you bake and cut into two perfect cakes. Now let's move on to the interphase, which is 90% of the cell's life. Interphase is the longest phase of the cell cycle, making up about 90% of a cell's life. Although the cell isn't dividing during this time, it's incredibly busy. growing, copying its DNA, and preparing everything it needs for cell division. You can think of interphase as the prep stage before the big event, which is the mitosis. It's like a student preparing for an exam, gathering materials, taking notes, and reviewing everything before test day. The three stages of interphase are first again G1 or growth 1, growth and function. The cell grows in size, it produces proteins and organelles it needs to function. This is when the cell is doing its normal job, like a skin cell helping to protect the body. At the end of G1, the cell reaches a checkpoint. Is the cell big enough? Is the DNA undamaged? If not, the cycle pauses. So for an analogy, this phase is like gathering school supplies and getting organized. The second phase is the S phase or the synthesis phase. This is under DNA replication. The cell copies its DNA so that each new daughter cell will have a complete set of genetic information. Every chromosome is duplicated, forming sister chromatids held together by a centromere. So for an analogy, it's like copying your entire notebook so you and a friend can each have one. And then the last stage under interphase is G2. This is where final checks and prep. The cell makes final preparations for division. It produces spindle fibers and other molecules needed for mitosis. The cell also checks for DNA errors and tries to fix them before moving on. Another checkpoint ensures everything is ready for mitosis. So for an analogy, it's like double checking your backpack before leaving for school. Why is interphase important? Of course, it ensures the cell has everything it needs before dividing. Cell cycle checkpoints help prevent mistakes, which could lead to cell death or even cancer if ignored. Only when everything is ready does the cell move on to the main event, which is the mitosis. Mitosis is the part of the cell cycle where a single cell divides into two identical daughter cells. These new cells have the same number of chromosomes and the same DNA as the original parent cell. Mitosis is how your body grows and repairs itself. It's the reason your skin heals after a cut, your hair grows, and your body can replace old or damaged cells. Now, when does mitosis happen? Mitosis happens after interphase, once the cell has grown, copied its DNA, and is fully prepared to divide. It's followed by cytokinesis, the splitting of the cytoplasm, which completes the division. Mitosis has 4 phases. You can remember the steps of mitosis with the acronym PMOT. The first phase is prophase. Chromosomes condense and become visible. The nuclear envelope breaks down, spindle fibers begin to form from structures called centrioles, which is found in animal cells. Chromosomes are now made of two sister chromatids connected by a centromere. Think of it like packing up your things before a big move. Second phase is the metaphase, middle lineup. The spindle fibers attached to the centromeres. Chromosomes are pulled and lined up in the center or the equator of the cell, like students lining up in the middle of the gym for a photo. Now, the third phase is the anaphase, which stands for apart. The centromeres are split. The spindle fibers pull sister chromatids apart to opposite ends or opposite poles of the cell. Imagine twins getting pulled to opposite sides of a room. And the last phase is the telophase, or two nuclei. Chromatids reach the pole and begin to uncoil back into chromatin. Two new nuclear envelopes form around the DNA. The cell now has two nuclei and is almost ready to fully split, like setting up two new homes after unpacking everything. Then comes cytokinesis. The cell physically splits into two daughter cells. Cytokinesis is where the cytoplasm divides, creating two separate cells. In animal cells, the membrane pinches in, called the cleavage furrow. In plant cells, a cell plate forms between two new nuclei. Now you have two genetically identical daughter cells, each with the same number of chromosomes as the original. This keeps everything consistent and controlled. Now, why mitosis is important? Of course, first for growth, from a single fertilized egg to trillions of cells in your body. It's also needed for repair, to heal wounds and replaces damaged cells. It's also needed for asexual reproduction. Some organisms reproduce by mitosis, like bacteria, through technically they use binary fission, a similar concept. But how does the cell know when to divide or when to stop? That's where cell cycle regulation comes in. Throughout the cycle, there are checkpoints like security gates that check if the cell is ready to move on. Here are the major checkpoints. G1 checkpoint. Is the cell big enough? Is the DNA okay? Second checkpoint is G2 checkpoint. Did the DNA copy correctly? The third checkpoint is the M checkpoint. Are the chromosomes lined up properly? These checkpoints are controlled by proteins like cyclins and CDKs, or cyclin-dependent kinases. If anything goes wrong, the cycle pauses or even stops. Now, why is this regulation important? First, of course, to prevent uncontrolled growth or for cancer prevention. If cell cycle regulation fails, damaged cells might keep dividing uncontrollably. This can lead to tumors and diseases like cancer. Many cancers are caused by mutations in genes that control the cell cycle, like p53, a key tumor suppressor. It also protects the DNA. Regulation ensures that DNA is accurately copied and passed on. If cells divide with damaged or incomplete DNA, the new cells may not function properly or may die. And of course, it maintains healthy tissues. Regulation helps your body grow at the right pace and replace old cells only when needed. For example, your skin constantly sheds dead cells and makes new ones. Regulation ensures it doesn't overdo it. Now let's wrap up with a quick recap. The cell cycle is how cells grow and divide. Interphase is where the cell prepares G1, S, and G2. Mitosis divides the cell into two identical ones. Remember PMAT. Checkpoints help the body control the process and prevent mistakes. Without regulation, the cycle can go wrong, leading to diseases like cancer. So just remember, every second, your cells are dividing with amazing precision. And now you understand exactly how they do it. Thank you for listening. See you in the next module. Bye-bye.